has holes in it, but still holds water? Sponge. Nicely done. Wow, that was fast. You've heard that before, huh? Okay. How about this one? You ready? Sponge. No, not a sponge. What do you call an alligator in a vest? An investigator. Wow. I might have to pull out another one because you guys are way too bright today. Let's see if we can stump you. What type of dance do teachers like the best? Freaky Friday? It's like this ink blot test. What is the... shout out the first thing that comes to mind when you see this picture. What type of dance do teachers like the best? Attendance. All right, finally I got you. I got you with the lame one. So, at this point, we're bridging the nervous system to the muscular system. And the two most important topics for 201 are right now, in this part of the semester. Action potential and nerve physiology, which we just learned about, and muscle contraction and muscle physiology, which we're going to learn about. And if you look at the amount of time that we spend in nerve physiology into muscle physiology and contraction, collectively over the semester, it's about a third of the entire course, or more. Just the number of test questions, the amount of 2 exams out of 5, okay, are over that content. Want to bet that 5th exam, some of the 35% of the review material is going to be over those 2 concepts. Okay, so if we were to hallmark this course on 2 major things, physiologically, they would be nerve physiology and muscle physiology. We talked about skin, we talked about bone, we talked about the different types of tissues and histology. Not that that stuff isn't as important, it's just by volume, it's not nearly as much. So we're really in the heart of the course right now. And then when we wind it down in our final unit with special senses, it's more details about nerve physiology. Because the special senses are receptor organs that fire action potentials. So if you look at really 3 exams out of 5 are spent on those really 2 main concepts. Okay? So I know it's springtime and it's super nice outdoors, but we're not done yet. And that includes me. I mean, I'm human just like you guys. So staying focused for the remainder of the semester when it's beautiful outside and the snow is actually still really good on the mountain is hard. Okay? The trails are nice. I know a lot of you guys like the outdoors, and the trails didn't get whacked this winter because we had a nice light winter in town and we still had reasonable snow up on the mountain. Hopefully it'll get really windy and miserable outside, so you just want to go in the library and study. Just kidding. But we still have to buckle down and try to follow through until May 6th. Okay?
May 6th is the last day of school?
That's the last day of school, in my opinion, for you, because that's my final. What you do on May 7th is up to you, but if you have a physics exam, you should probably show up. Okay? So here we're in the peripheral nervous system, and I want to back up a little bit before we get into our review material, and I want to talk about where we are. Okay, if this is the heart of the material, I don't want to go too fast, too quickly. I want to make sure that we orient ourselves. We just had an exam. We're covering content we didn't quite get to, and it's supposedly related to where we're going, he keeps saying. But let's see where we've been. So we talked about the central nervous system, and we didn't quite get through all of the spinal cord, and that's fine. Because the spinal cord, remember, we put these categories, relatively arbitrary categories, because they tend to fit into these nice categories. The brain and the spinal cord make sense to call it the central nervous system. It's the central part of the nervous system. But then we sort of make an arbitrary division, say anything outside of that, like cranial nerves or spinal nerves, are not considered part of the CNS. They're part of the periphery. Well, when we get out into the peripheral nervous system, does the body care that you call it CNS or PNS? Not really. But when we get out into the periphery, which is where we're going to be before we get to muscle, which makes sense because the muscles are in the periphery. So we're sending the signal out through the spinal cord, and it's going out to muscles, glandular organs to do things. We're also going to be receiving input from the periphery, or the peripheral nervous system, on the sensory side. So our motor output to do stuff is over here on the motor division. And we can further divide that into somatic and visceral. Remember, the visceral is the autonomic. We've already covered this. And the autonomic is further divided into parasympathetic and sympathetic. And we can fly in this secondary terminology for the autonomic nervous system, which just overlays this visceral, because it's two names for the same system. Well, if we back up to the sensory, we also see that we have automatic and we have other body functions. Somatic means any body cell, anywhere else in the body. Visceral or autonomic, as we can see over here, is it's referring to those things that you don't have to think about. So we're going to talk today, first up, about how the spinal cord is further organized in more detail. Then we're going to talk about some reflexes that take place. Okay? Then we're going to talk about some autonomic reflexes. And then we're going to move into more details in the periphery as we dive into our peripheral nervous system. Okay? If we look at what the peripheral nervous system does, specifically our autonomic division, or our visceral side, the impact across the body is vast. It's extremely broad. You can see all the different representative organs that are shown here. Some of them we've covered, many of which we haven't because you're going to get to them next semester in 202. But you can appreciate that this list of things that the visceral nervous system does are unconsciously controlled. So it modulates homeostasis. Modulates homeostasis. A lot of questions on the last exam were this. What does the word compromise mean? Do you guys remember the word compromised on the exam?
No.
No? The word was there because we had the SIs and I had lots of questions about the word compromise. So, I mean, if you have a question on the exam about the definition of a word like that, you can ask. Part of this class is to try to expose you to a broader vocabulary. So, what does modulate mean? What's another word for modulate? One that's not on the screen. Regulation. Change. What else? Give me some synonyms. Synonyms. That's a hard word to say. Synonyms. Is that better? Synonyms. We're going to increase your vocabulary with synonyms. What's another synonym for modulate, regulate, control? What else? What's that? Governs. Okay. Oversees. Guides. Influences. Did we say that one? So all of these words mean that it controls it. It determines what's going to happen to it. Look at the functions that this has influence over. And this is the category that we're about to move into and talk about. Body temperature, digestion, heart rate, metabolism, breathing, defecation and urination, as well as pupil diameter. All of these are things that aren't voluntarily controlled or modulated. Can you override and influence them? Yes. Okay? But the autonomic nervous system takes care of it without you thinking about it. Do you have to think to breathe?
No.
No. Do you have to think to create a heartbeat? No. In certain circumstances, can you override that control with a somatic control? Yes. And that's where the nerve energy comes from. That's why it's important to have these action potentials. Question?
I want to say, I think that defecation and urination are somewhere in like a no man's land, because you can hold both poop and pee. So I think they're kind of like in a no man's land. I don't think they're
Thank goodness, right?
Yeah.
Oh, whoops!
Yeah, they're neither one. They're in the middle. They're in no man's land.
Yeah. Yeah. Well, so they're not in no man's land, and here's why. It's a good question. It's a curiosity, right? So the control or the influence is when the stimulus shows up, the activity happens. Okay? And then at a young age, you're trained, okay, by usually your parents, if not, then your peers. Because they're like, dude, you shouldn't, like, do that. Okay? So I tell my friends, like, damn, my son keeps picking his nose. Don't worry, it'll eventually work itself out in grade school. Right? Like, I don't know why she won't talk to me. Well, if you got your finger out of your nose, right? So you can override those things. So they're not necessarily no man's land. They fit under autonomic, but they're a perfect example of how we can override or control them. And we're going to talk about them in more detail. Okay? All right, so let's now pick up where we kind of left off with the spinal cord, because the spinal cord allows us to interact with that peripheral nervous system. It's out near the periphery. It's not part of the periphery, but it's near the periphery, so it has activity out there. It's organized in a very systematic way. Okay? It's almost like it had a label maker when it was being designed. I love label makers. How many of you guys like label makers? Who has one and uses it regularly? How many of you have labeled your label maker? Yeah, you might need help if that was you. If you raise your hand, you're like, whoa, yeah, all right. So we have gray matter, we have white matter, and these guys are organized. You can see here, the gray matter exists in the center portion. This is our central canal as it comes down. So you're looking through a slice. In the front is the anterior or the ventral side. In the back is the posterior or the dorsal side. So you're looking at a cross section and you're looking top down. And as you look top down, you can see that there looks like this butterfly pattern. Right? It's very characteristic. And this butterfly pattern is the gray material. Okay? It's unmyelinated. And you have an anterior portion, an anterior horn, or a ventral horn. It's these two structures that come down, or it looks like an H. And you have a posterior horn, or a dorsal horn, which is towards the back. And information that's going to come in, it's going to come in from the periphery, from the dorsal horn, or from the posterior horn, and that's sensory information. So if you remember when we were in the brain, we said that the brain was organized in one axis of back to front. And it was sensory information towards the back and motor output towards the front. Well, this component of the central nervous system is organized in the very same way. It makes it nice and easy. So the posterior sensory input comes in. And as it moves out, it's under motor efferent control. So motor efferent nerves come out the front, or the anterior, or the ventral root horn. Now we have some mixture up at the lateral horn where you've got a little bit of both that take place. The lateral horn is not shown because it only exists in a very specific segment of the spinal cord. That's T2 through L1. Okay? And it's really in this region right here. Okay? But things up top and things down low are organized in this pattern right here. Now our white matter is everything outside of that butterfly shape, or that H. And the white matter is made up of myelinated tracts. It contains myelin. They're composed of bundles of axons. And the tracts usually carry information from one part of the central nervous system to another. If you notice, the gray matter takes information that comes out and puts it back out into the periphery. This is going to be, the gray matter is the loop that we use for our reflex arcs. The white matter is organized in these columns, and these columns go up and down. They go superior and inferiorly. And as the columns are organized, you're going to still see the columns that are considered ascending tracts. And you can see here is a collection of columns in red. Here's one, and those are going up, and they're going up to the brain, and they're taking sensory information via afferent neurons up to the brain. And then what comes down these descending tracts, which are shown in the green, and it's not perfectly delineated. Right? If you put a line right through here, you can see that, okay, the green ones are more preferentially organized towards the anterior aspect because they're motor output. And the red ones, or the sensory, are more posteriorly organized because they're sensory. But it's not exactly 50-50. You with me? Like if you cut right through here and say, oh, well, Keller said it was sensory information towards the posterior aspect and motor information towards the anterior aspect. It's still true, but you kind of see it's more of a trend when you're talking about this white matter. The gray matter is hard and fast. The white matter is a little bit more fuzzy, if you will.
The white matter is all the stuff around the brown H, right?
That's correct.
Okay.
That's correct. And if you notice, like the reason that we've separated it on this picture, what about this right here, the mirror of this one? Is there a ascending tract on the left side? Absolutely. Okay, so there's an ascending tract on the left side, there's an ascending tract here and here. You see this green circle, which is a descending tract on the left? There's actually a space for it right over here where it exists. You guys following? So you're sending information from both sides of the body up and down. But for clearness in this picture, or to delineate ascending versus descending, we're just showing one side in red and the other side in green. Now, yes sir, in the Kings.
Just when you try to understand the lateral horn, that exists because of the sympathetic nervous system?
Yeah, so the lateral horn handles lots of the sympathetic nervous system innervation. So that's what it's responsible for. Okay, but you don't see it in the standard preps, because we take these preps at more cervical or lumbar locations for simplicity, so we don't complicate things. Okay. So the main focus that I want you guys to hone in on is the organization of the dorsal and the ventral horns. Okay, for the most part, you can kind of ignore the lateral horn. I won't ask a test question about it. Okay. So if we look at organization of the nerves and the architecture, we're going to see some themes that exist, architectural themes. So over on the far right is an electron micrograph. This electron micrograph is showing this architectural bundle. So the analogy that I like to use is if you took a group of pencils or markers, right, and you bundled these guys together, or if you took a bundle of well, really just about anything cylindrical, okay? And then you wrapped it with a sheath, and you look at it on end. You see that view that you're seeing there? That's that view that you see on that far right image. So you're seeing a cylinder, and you're looking at it on end. Okay, and as you go to the cartoon now, and you look at these nerves, the way these nerves are packaged and organized, it's kind of like a bundle of pencils or markers kind of tied together. And they're tied together and they're wrapped with a sheath instead of a rubber band. Okay? So if we go down to the smallest, finest detail at the individual nerve fiber, which is a neuron. A nerve fiber is another word for a neuron. Here we've got unmyelinated, and you can see them and appreciate them in just plain yellow. And then this one actually has myelin around it. And around that myelinated fiber, you see what we call an endoneurium. So the endoneurium, endo meaning inside, is the wrapping around the nerve fiber. And this endoneurium that surrounds it, you put a bunch of these fibers together in this bundle, and then you wrap it with this perimeter wrapping, perineurium. And then you get a group of these bundles of nerve fibers together, and now you're getting to a space or a size where you need to think about perfusing them with nutrition. Because, remember, nerves need oxygenation. Proof of that is if you sit on your leg too long, or you cross your legs and you don't move, and you cut off blood flow to those distal nerves, they start yelling at you. They yell at you with that pinprick feeling, which is called parathesis, telling you, I'm starving from oxygen depletion. Move your leg. Okay? So here we get a group of these together, and we wrap them in this epinurium. Remember, epidermis, the outermost surface covering. So the epinurium is this outer wrapping of these individual wrapped perinurium bundles that contain myelinated and unmyelinated nerve fibers. And this is how the nerve, this is one nerve coming out. Okay, all the different constitutive parts. Now, if we go back to bone, bone was organized in a very similar strategy, and these were osteons. Remember that?
Yeah.
When we move forward into muscle, we're going to see muscle fibers organized in much of the same shape. You see this all over biology. You see motifs, architectural motifs that mimic each other. Because if they work, they're used again and again and again. Okay? How many of you have a car? Or a bicycle? Or a skateboard? And the wheels on those objects are in what shape? Roundish, right? Amazing. It works. So we stopped using square wheels. Okay? So you see this motif exist in bone organization, in nerve organization, and you're going to see it again in muscle when we get there. Question?
Oh, okay. I'm wondering, the holes, the fossa, and the bones of the vertebral column, is this what goes into them in order to get to the spinal cord?
Yeah, so these things come out of those foramina or fossa, either ones, depending upon where you are, and that's why they exist. Anytime you have a hole in a bone, usually a blood vessel or a nerve is coming through it. That's why they exist.
That's what the holes in the vertebral bones are for.
Yes.
Okay.
Yep.
So the epinurium, it wraps around the fascicle, or all of the...
No, there's one individual fascicle is this. This is a fascicle. So you got a bundle of fascicles together and you surround them with this epinurium. Okay? All right, so if we look at how the tracts exist in the spinal cord, and now you can start appreciating how we're going to connect the brain to the rest of the body. That's why we're spending time to do this. Here's the brain up top. And you can see some features as we work our way down. So we're going to send a signal up to the brain. These are actually spinal tracts that are ascending tracts. Okay? So let's start at the bottom of the slide. Here's how to read this slide. Start at the bottom and work our way to the top. First off, what you'll notice that we've already talked about is receptor information on the right side of the body ends up on the left hemisphere. Does it tell you that? Yeah, see? Information comes in here. I also told you it comes in on the posterior aspect because it's sensory, and then it shoots up. It shoots up these columns, and they make their way ultimately to the level of the medulla. So here, at the level of the spinal cord, what comes in are receptors for body movements. They come in the dorsal root horn, and they leave via a first-order neuron up. They stay on the same side until they get to the level of the medulla.
And that's where it switches direction.
At the level of the medulla is where it crosses over. As it crosses over, it continues its way superiorly via a second-order neuron. You can see it labeled right here, second-order neuron. And this crossing over, that term is called decussate. Decussation. So decussation is a crossing over of a nerve signal from one side to the other side. As it crosses over, it goes to what we call the contralateral side. Contralateral is the opposite side. If it stayed on the same side, we'd actually call that ipsilateral. So ipsilateral means the information comes in on the right, and it would stay on the right side and go up to the right hemisphere. You say, well, that never happens. Well, there are cases where we get what we call hemidecussation. Hemidecussation is like a half crossing over. So half of the signal goes to the contralateral side, and half of the signal stays on the ipsilateral side. And the example that we're going to talk about in a couple of weeks is with vision. When you see something with either eye, okay, you can close one eye or the other, you're getting that image coming in, and it's going to both lobes. So the left eye doesn't just bring in information and send it to the right portion of the occipital lobe. It goes to both, and vice versa. The right eye sends it to both lobes. That's called hemidecussation.
Is
So
that the only exception? Vision?
it's the only one that I know of. Yeah, off the top of my head. So at the level of the midbrain, you can see we're starting to send information up. Eventually we get into the thalamus. Remember, the thalamus was a relay station. The thalamus was information coming in. I'm going to decide where I'm going to send that signal that's coming from the periphery. And as it goes out, this one's showing the third order neuron leaving the thalamus, going to the postcentral gyrus. Postcentral gyrus is towards the back. Right? This information came from the right side. Now it ends up on the postcentral gyrus towards the back, like right about here. And it's saying, ow, I just stepped on a tack. Okay? Then you're going to need to do something with that foot and move it. So you have to interpret that signal, send it via interneurons to a motor track, and we need to go down a descending pathway, which is a motor output or efferent tracks. So if we do a descending pathway, we're going to start at the top and we're going to go to the bottom now. So if we start at the top, we can start on either side. Right? You can start on either side. You can see if we start on the patient's right side, we're over here. Patient's left side is over here. Remember, we're in anatomical position, so it's like facing you. Now, the motor cortex, which is the precentral gyrus, is going to send motor efferents down. They make it through the midbrain. They eventually get to the medulla. At the level of the medulla is where they cross over again and they decussate. So the nice thing is the medulla is the decussation point for both ascending tracts as well as descending tracts. It's convenient. But it also tells you something important about that organ, that it helps to coordinate those activities, and it does it in both directions. Now you can see these columns. We're in green, and we're going downwards, and we're towards the anterior aspect or the ventral aspect, and we are eventually going to make it out, depending upon which side the signal came from. If it's from the left hemisphere, it'll go out to skeletal muscles here. If it's from the right hemisphere, it'll go out to skeletal muscles on the left aspect. Okay? So ascending tracts go up, decussate at the level of the medulla. Descending tracts come down. Again, they decussate at the level of the medulla. And they help to coordinate these limb movements. And question? Or?
Yeah. Is there an advantage to decussating, or is that just something that, like, evolutionarily just happened?
It's a great question, and I don't have an answer for you.
Okay.
We haven't. I've never seen any really good explanation as to why have the information cross over. The reason that it hemidecussates in vision makes a lot of sense, because there's redundancy there.
Right.
So if you're trying to get away from a predator, okay, or if you're trying to catch your meal, and something happened to you in the field, and you lost vision in one eye, it would be nice to be able to compensate and still be able to figure out, without having true binocular vision, where exactly is that prey or where is that predator. But with everything that we do, having it go to the other side, I don't have a good explanation for you. I wish I did, because we get that question a lot. It would make more sense if it hemidecussated to say, oh, well, you have two brains and you have a backup redundancy on either side. But that doesn't happen. It only happens with vision. So it's a good question. Okay?
I have a question.
Yeah.
If you get injured and you can't fully decussate somewhere, does it work in the sense of other things where your brain will be organized
so neuroplasticity.
Yeah.
So the problem is, at the level of the medulla, if you had an injury at the medulla, you probably wouldn't make it, survive, because you're so far down on the brainstem that you're dealing with a lot of autonomic functions. And so if you had an injury here, much of the stuff below would be actually done. Okay. Now, with medical advances, we could probably keep you alive. You'd be on a respirator. You'd be in a wheelchair. You probably wouldn't have control over your autonomic functions like micturition and defecation. Micturition means urination. But again, medically, we can hook you up to technology to allow you to have a quote-unquote normal life. But out in the field, for the question that was asked earlier, if we talk about before this is how we lived, that organism probably wouldn't make it. Okay?
For somebody who is paralyzed, do they still have ascending tracts even though they wouldn't?
Yeah, it depends on if it's a complete spinal cord severing, the question is if somebody had a spinal cord injury, could they have ascending tract still, or could they have some descending? It depends on where the injury is. If it's a complete severing in the spinal cord, then you wouldn't have either. Okay? But if it's partial severing or partial damage, you may have feeling, you may not have motion, or vice versa. Okay. I want you to be familiar as we work through these tracks. You can kind of appreciate every single one of these that we've highlighted. We actually have names for them. And they do certain things. Now, this is level of detail that's far beyond the scope of this course. Okay, so another way of saying that is for you to sit here and memorize all of these tracks is not necessary. What I want you to be able to do is know, if I gave you a question about sensory input, I want you to know which way does it go. Where would that track most likely be? Where is it going to ultimately end up in the brain? If I told you what center it actually connected to after it went to the thalamus, you should be able to tell me what's it responsible for. I want you to tell me where does it go when it comes down. Where does it innervate? What track will it probably follow, on the anterior or the posterior aspect? Do you need to be able to tell me, no, it's exactly the medial reticular spinal tract, right? No, not necessarily. Okay? Those of you that aren't here won't get that information unless they watch the YouTube lecture, unless you tell them. Okay? But I want, for sake of completeness, to put this slide up here so you don't say, oh, well, he just like painted these areas and said, you know, it exists on the other side. No, these are very specific known regions in the spinal cord. We've actually put probes into the field, not me, put probes into these regions and can register information coming from sensory structures, or we can register motor output. In fact, you can actually make the organism behave based upon where your electrophysiology probes are located. So these are actually very well described. And I think that's fair, that you need to know the general organization and not necessarily all of these specifics. Now, as we work down through the spinal cord, the whole point of today's lecture is to get us to the periphery and then start covering information in the periphery. So as we come down and we go out into the periphery, once we leave, now we're into what we call plexi. So we're actually leaving the spinal cord at this point. We'll probably come back to it here in a moment, especially when we talk about reflexes. But we have to go out so you can see the whole track in the process, and we can come back and say, okay, now what does a reflex look like? So these guys are named, it's singular plexus, plexi is plural, and a plexi is a network of nerves, or it could be blood vessels, right? Like, for example, we talked about a structure known as the choroid plexus. Remember that one? The choroid plexus is a network of blood vessels that exist in the ventricular system of the central nervous system. Plexus is a network of nerves here at the level of the periphery. Network of intersecting nerves, and they typically combine sets of spinal nerves that serve the same general area of the body into one large group nerve. So for example, you're going to find them in areas like the cervical plexi, the brachial plexi, the lumbar, the sacral, and the coccygeal. Now, the thoracic is organized a little differently in the sense that we have thoracic nerves. You can see them on this picture. But they don't, you see how they're individual? They're individual, whereas these guys are intersecting. So a plexus almost looks disorganized. And in the thoracic region, T1 through 12, it's very, very systematic. One nerve coming out. So it's not truly a plexus. There, unfortunately, a lot of students walk away and go, oh, there's no nerves in the thoracic region. Okay? Because there's no plexus. No, it's not a plexus because, do you see how this is shaped? How it's kind of like all over? And these guys are all nice and organized. And then it kind of gets all squirrely again down here in the plexus.
So plexi can only refer to collections of either blood vessels or nerves. Are the only two things that can constitute a plexus.
Yeah, I mean, you could probably use it to talk about roadways in Europe, but people might look at you weird, But yeah, I mean, nerves and blood vessels are the two examples that we see that exist in the body as plexi.
Okay.
Is there a reason why the thoracic nerves aren't in plexi? Or is it just because they're not going
The reason is they like to follow the pattern of the ribs in that region. And so you can see they almost look like ribs. And so you have a little bit more organizational structure there. Whereas up here, there's no bones guiding their direction. And right here, like the level of the waist, underneath your rib and before you get to the hip bone, or the tip of the os coxa, you have a lot of extra space. So those nerves don't have any directional pattern. Nerves like to follow structures during development when they grow. Blood vessels do the same thing. Okay? That's good. It's a really good question. If you look at this picture, it almost looks like these are ribs coming out. And the nerve is following the architectural shape of the rib. Okay, so here's a possible test question. Which analogy is the best comparing axon to endoneurium? Axon to endoneurium. Who has an answer and why?
I'm going to say compact bone periosteum.
Compact bone periosteum. Why?
Well, because the periosteum is like the thing that surrounds the... So they're kind of like analogous structures.
Yeah, they are. Yeah, that's actually correct. Okay, so the periosteum covers over the compact bone, similar to the way the endoneurium surrounds the axon itself. Endo inside, right? The epineurium is on the outer portion of the fascicles, but this is that analogy that I was talking about. I could put one on muscle as well, because we'll eventually get to muscle. Question? I made you forget because you had to wait too long.
Oh yeah.
Okay.
So are they just nerves if they come out all nice and orderly fashion, but they're plexi if they kind of come out and branch together?
Yes.
Okay.
Yeah. So the question is, if they come out all singular and streamlined, are they nerves? Yeah. If they come out like kind of all branching and connecting and network, then they would be considered a plexus. Yes. It's just called, they're just called thoracic nerves. You don't typically use the terminology plexus to refer to the thoracic nerves. You just call them thoracic nerves. Guys, it's a total nomenclature thing. I get it, but it's important to understand because if you're talking about a motorcycle accident patient, right? Let's go to our doctor role play. Remember the herpes?
Yeah.
Yeah. So now, you come in and a patient has a motorcycle wreck and they say there's damage to, the thoracic region and you're worried about damage to thoracic nerves. You understand what they're talking about. Or they say it's in the cervical region, it was a rear end or a rollover and they're worried about cervical plexus. You understand, we're still talking about nerves, but we just, maybe in the cervical region, we don't really know which region specifically, because now we've got kind of a network of nerves innervating. We're queuing up to reflexes.
So So if you damage one of the intersecting plexus nerves, will it damage like the other ones?
It depends. The question is, if one nerve intersecting plexus gets damaged, does it damage them all? Not necessarily. And that's one of the thought processes around having this plexus, is you get kind of a network of nerves that can serve an area. But the other problem is, if you have inflammation or irritation in the area, it can be more broad-based. Right? It can be really, really painful because you have more nerves per unit volume of tissue that could actually be affected. So if you have, for example, lower back pain, and it's shooting leg pain down your nerve, and you treat kind of as you go, it may take a while before you figure out where exactly to treat. Reflexes. Reflexes are the way that we automatically interact with our environment. And the first question that I want to pose before we get there is this. The left post-central gyrus receives information about the texture of the fabric you're touching with your left hand. Along which spinal nerve did this signal enter the spinal cord on its way to the brain? So think about it for a second. Think about it for a second. So I want someone to explain it before we share what the answer is. Raise your hand if you have an explanation to your answer before you tell us what your answer is. I want you to explain how you got to your answer before you tell us what your answer is. Okay? Who can do that? Up against the wall, please.
Well, the sensory is anterior, correct? Or is more?
Sensory is posterior.
Okay. So it would come in on A because it's on the right side, and it goes to the left central gyrus. So we posterior since it's sensory, and right side because it's on the left.
Perfect. Now, how about the second half of the question? Who can do that? It's going to go to the hand to drop what you're holding. And which pathway does it take? Everybody understand the question? I want the second half of this loop. The answer to the first part. Okay. The answer to the first part is A. Because the information ends up on the left hemisphere, so it has to come from the right side. And it has to come from the posterior or dorsal aspect because of sensory information. So it's going to be A, not B. Now, if we're going to send information from the left hemisphere to go out to the right hand, where is it going to exit? It's going to exit B. You guys catch that? Okay. All right. Does that hurt a little bit? It hurt. Okay. I can hear it moving. Is it? So let's talk about parts of a reflex. We have four main characteristics of a reflex. Number one, they're stereotypical. Number two, they're rapid or quick. Number three, they're involuntary, meaning you don't have to think about them. And number four is they need stimulation or they respond to sensory input. Stereotypical, quick or rapid, involuntary, and they respond to a stimulus or excitable. Now, on this particular slide, we've got a lot going on. So let's talk about what's happening in this slide before we get to two examples at the level of the muscle. Because I'm going to share with you four reflexes today. Okay? This is setting up the first one. This structure here is found within the muscle, and it's called the muscle spindle. We're going to characterize it in more detail here momentarily. But it is essentially part of a reflex. It's the sensory organ in a muscle that tells you about what's happening in that muscle. So for now, let's just pretend that this muscle is stretched. This spindle is activated. This structure here is blown up in higher magnification, and then we cut open this capsule so you can kind of peer inside of it. And that's where our next slide is going to start. Now you've got this yellowish neuron, which is your afferent neuron, which is coming in, and it's coming in on the dorsal aspect. That's your sensory afferent information. It gets to the dorsal side, and you can see there's a neuron that comes here, one that comes here, and then one that goes up to the brain. So it innervates, this sensory afferent innervates onto 3 separate neurons in this example. 2 that look like they're coming right back out at the level of the spinal cord. It comes in, and it comes right back out. And it's going to come out on the ventral aspect, so what do you know about it to be true? If it's on the ventral or the anterior aspect?
It's sensory.
No. It's motor. It's motor. Very good. Okay? So, simultaneously, that signal is sent up to the brain. So when we back up to our last unit, we talked about spatial summation. You guys remember that? Try not to. Spatial summation was where you're getting nervous input from different neurons onto one neuron. Right? We also talked about converging versus diverging circuitry. This is an example of diverging circuitry, right? Because information comes in and it separates the signal out. It's like a splitter. Right? It's like, if you have a cell phone charger and you have 4 cell phones and only 1 charger, you need like a splitter to split this signal. So here the signal comes out and it goes up. And as it comes out, you can see these are motor efferent pathways. One of them, the red one, comes right back to the same muscle. One of them, the blue one, goes to the antagonistic muscle. So in this case of the muscle spindle, when it comes back, if this muscle is being stretched, okay, extended, then the red one causes it to fire and contract. So it doesn't stretch out too far. This blue one goes to the antagonistic muscle to say, inhibit it, relax it. Because I don't want you opposing the flexion of this muscle, because I'm actually wanting to contract that particular muscle. So in this example, the red muscle is being shown and diagrammed as what? The quadriceps group. And the hamstrings are the blue innervated muscle. Okay? So they're antagonistic muscles, and if you want to contract and therefore extend the knee, you want to make sure you're relaxing your knee flexors. So this is a reflex that happens as I walk across this room right now. This information is happening so that I can move, and so I'm not like, I can't figure out like if I'm contracting, or it does it in synchrony. That's why you need dual signals to go to both muscle groups to tell them relax, contract, relax, to allow you to do this nice fluid motion. At the same time, there's information that goes up to the brain. And the information that goes up to the brain goes to the thalamus, the relay center, and it goes to the sensory cortex, the motor cortex. Right? The motor cortex says, oh, stop, there's something there, you should pick it up. So now you're able to stop, pick it up, because you can override sort of this natural pattern of walking. Okay, here's a terrible example, but you won't forget it. Have any of you guys seen like a movie or where a chicken gets its head cut off? and it can just like run around. So this will run on its own, okay, for a period of time until the ATP wears out, right? And the sodium potassium pumps can't work anymore, now you get no more signal, and then this reflex no longer happens. So you and I use these reflexes all day long to do normal routine events that we're sort of programmed to do. And we've gotten really good at using these particular reflexes. I want to finish out muscle spindle, and then I'll get to your question. So if we blow up the muscle spindle in higher magnification view, this is an organ, it's a reflex, it's an organ, a sensory organ that's found inside the muscle. All muscles have them. It gives us information about changes in the length of the muscle. It's a protective mechanism so that the muscle is not overstretched. It's consisting of intrafusal and extrafusal fibers. The intrafusal fibers are mostly the afferent sensory fibers. But you can see we've got afferent primaries, we have afferent secondaries, we also have gamma motor. So you can motor control the spindle from the inside. And then the fibers that exist on the outside are all motor, alpha motor fibers. So what do these intrafusal fibers do for us? Well, the primary afferents respond to the rate and to the amount of the stretch. To the rate and the amount. And the secondaries only respond to the degree of the stretch, or the amount of the stretch. So you've got primaries that do both rate, how fast, and how much. And secondaries that just deal with how much. Then the gammas adjust and maintain the sensitivity of the muscle spindle. So these can be calibrated to your particular body and to your particular training level, to your particular regular motor patterns. Our extrafusal fibers. These guys are stimulated to contract the whole muscle. That's why they're outside of the muscle spindle, because they need to innervate the muscle tissue itself and cause an activity to those skeletal muscle fibers. We're going to skip a slide that you might have, which is called flexor withdrawal, or maybe I deleted it, I can't remember. And we're going to move to our second example at the level of the muscle. This second example is called the Golgi tendon. So it's very similar to the spindle, and then it's giving information about the muscle to the muscle, but it responds to muscle tension, not to changes in length. So the muscle spindle is about stretching the muscle. This is actually what's the tension, or the force over a length. That's what tension is. And it can be caused by either passive or it can be caused by active muscle contraction. You're going to find these Golgi tendons within this connective tissue. As if you see this is at the level of the femur that we're looking at here. And here is one of these large thigh muscles that comes in, and this little rectangle is the blow-up of this segment. And where the tendons insert into the bone is where you're going to find a Golgi tendon organ. It makes sense they call them, it's a funky name, right? Golgi tendon organ, like, ooh, well, that seems weird. But it's called that because it lies within the tendon, and it's an organ itself. It's a tension organ. So these are considered proprioceptors within the tendon, and with excessive tension, excessive force in the muscle, they actually inhibit the motor neuron so that you don't overwork that particular muscle. How do these guys work? Well, here's a little diagram that kind of explains this loop. So when you look at the spinal cord slice, remember I told you by the end of today's lecture, I want you to see how we connect our central nervous system to our periphery. That's the whole goal of today. And then we're going to fill in all the other details as we move on. So if you see our spinal cord slice, here's our Golgi tendon in light blue. Let's start there. This is our quadriceps, or our quads muscle group, that are knee extenders. When they contract, the leg goes up, right? So when this thing is being contracted and you sense with your tendon organ that the force is getting pretty high, it sends a signal, an afferent sensory fiber in the posterior horn. It innervates onto two, just like we saw with the spindle, two interneurons. These interneurons, one goes out via red and goes directly back to the organ and is going to block it, right? The other one goes here to the antagonist and it actually stimulates it, right? Why does this stimulatory, you know it's stimulatory because it's a positive, the purple is positive, stimulating the hamstrings, the flexors, and the negative, the red one, comes to the muscle that's being contracted and inhibits it. Why is it doing that?
Is that because the antagonistic muscle that's stopping and reversing the action of a potentially dangerous situation?
Right. So it is another protective mechanism, but here's how it works, right? If you didn't have it in the swing phase, when you're walking, it wouldn't be quite as smooth as we walk, right? There's these nice smooth pattern switches between contraction and flexion. We're not like, right? Like, so it can, and if you throw your leg out like that repetitively, you'll do damage to the muscle, you'll do damage to the tendon, you'll do damage to the joint. Okay, so just think, our nice fluid walking that we enjoy of contraction and relaxation, it isn't, this, awkward throw where we're contracting these muscles here to extend, but we don't, kick a soccer ball every time we walk across the front of the classroom. Okay? So these two organs work together to give us these nice fluid motions. The last two that I want to cover today, I'm going to take you to two videos. The videos are embedded. I think we'll get through them. Stay with me. We'll have a nice stopping point. I'm going very slow, purposefully, because I want to transition well into the periphery. I don't want to just jump into it. These are two more that we're going to talk about. Visceral autonomic reflexes. They're unconscious. They're autonomic. These guys are stereotyped. That means they're the same every time. They're slower than what we just came from. And they're completely involuntary. The first one that we're going to look at is our baroreceptor reflex.
Increased blood pressure stretches the carotid arteries and aorta, causing the baroreceptors to increase their basal rate of action potential generation. Action potentials are conducted by the glossopharyngeal and vagus nerves to the cardioregulatory and vasomotor centers in the medulla oblongata. As a result of increased stimulation from the baroreceptors, the cardioregulatory center increases parasympathetic stimulation to the heart, which decreases the heart rate. Also, as a result of the increased stimulation from the baroreceptors, the cardiovascular center decreases sympathetic stimulation to the heart, which decreases heart rate and stroke volume. The vasomotor center decreases sympathetic stimulation to blood vessels, causing vasodilation. The vasodilation, along with the decreased heart rate and decreased stroke volume, bring the elevated blood pressure back toward normal. If the initial problem were a decrease in blood pressure, the activities and effects of the baroreceptors, cardiovascular center, and vasomotor center would be the opposite of what is illustrated.
Okay, so just like what we talked about, at the level of the muscle, we have reflexes that occur in other parts of the body. Okay? And they're used to control all sorts of other important functions, like blood pressure and how fast the heart works. Okay? So, we've got Golgi tendon, we have muscle spindle. Now we have this baroreceptor reflex. We're going to look at a fourth one right now, which is the mituration reflex. And this is how we control peeing. Defecation works much the same way. Okay, so they are reflexes, but we can override these reflexes as well. Right? Can you override the reflex that I was just illustrating with the walking? Of course. Can you override this mituration reflex? And it's some people sometimes, right? Of course. So let's take a peek at this last one.
Mituration reflex involves impulses traveling from the urinary bladder to the sacral region of the spinal cord and from the sacral region of the spinal cord back to the bladder. It is coordinated by neurons in the spinal cord and can be influenced by signals from the brain. When the urinary bladder becomes stretched, there is an increase in the frequency of action potentials carried from the bladder wall to the sacral region of the spinal cord. In response, parasympathetic neurons from the spinal cord to the bladder are activated, and this causes the smooth muscle on the bladder wall to contract. The sensory signals to the sacral region of the spinal cord also stimulate ascending pathways to the pons and cerebrum, which results in a conscious desire to urinate. If urination is not convenient at the time, the brain sends impulses down the spinal cord to inhibit the mituration reflex.
This is the training.
Impulses carried via somatic motor neurons keep the external urinary sphincter contracted, which also prevents urination.
That can be
When
facilitated by
urination
this
is
one.
desired,
Right?
signals from the brain stimulate the mituration reflex. The brain also decreases action potentials in the somatic motor neurons to relax the external urinary sphincter.
Okay, so stay with me as we wrap up. We have the baroreceptor reflex, we have the mituration reflex, we have our Golgi tendon, and we have our muscle spindle. So there's 4 that we've covered today. Now we are officially connecting our central nervous system to our periphery, and you have at least 4 examples of how that is done. So when we come back on Wednesday, we're going to pick it up here, and we're going to get into the autonomic nervous system in a lot more details. I encourage you to read this material for this remaining portion of the lecture and lecture 2 that we're going to be covering on Wednesday. Otherwise, you're going to be lost. Okay? Okay. See you Wednesday.